Hi everyone, welcome to my channel Biology, and I am Ga3 and today I come with another topic, or you can say another video, that is the gibberellin hormone. This is one kind of the plant hormone, and before explaining about this, I want to say about two or three actually plant hormones I already discussed in my previous videos. You can check that one, okay? And now I want to say obviously thanks to all of you who have subscribed my channel and support me for the growth of my channel, okay? So let's start today's topic, that is the gibberellin hormone. What is the gibberellin hormone? Gibberellin hormone, actually these are the growth regulator or plant's growth regulator. Actually it helps in the plant's growth. So generally its function, generally analysis, after analysis this gibberellin's function, it is seen that the internode system of the plant, you know, all the, there is the node system. In the stem there are nodes and the internodes. So where the gibberellin's functions actually activate, or where the gibberellin's functions, or which part, on which part the plant's body gibberellin actually functions. So it is seen that there is an internode system in the plant stem. Okay, the two parts, two nodes, there are two nodes. Between the two nodes there is a, so you know, it is called the internode system. So the meristematic, how the plants grow, due to the cell division that occurs in the meristematic tissue, and where we see the meristematic tissue, either it is seen in the plant's apex region or it is seen in the intercalary system. Okay, that means the between the two node system. Okay, between the two node, that means the internode region, there is actually the meristematic tissue. So if the cell division is increasing in the apical part, and this the meristematic tissue of the apical part, and this intercalary system or intercalary meristematic tissue, then the plant's elongation is seen. So actually gibberellin, this actually functions here, this part. This help to increase this internode part of the plant's body, and in that way the elongations of the plant's body is seen properly. Clear? So you just here you just know that what is gibberellin actually is one. It is actually, these are the group of the tetracyclic diterpenoid component. Okay, tetra means four, tetracyclic diterpenoid component, and generally made up of four isoprene units, okay, and with int gibberellin ring. One ring is there, this ring is called the int gibberellin ring, and four isoprene units, they attach to each other during the formation of the gibberellin. Okay, and it is first discovered from the fungal disease of rice, and the pathogen was the Gibberella fujikuri. Okay, Gibberella fujikuri. So this is actually gibberellin hormone, and its activity, obviously its function I will discuss after that. First I discuss its biosynthesis, or how it synthesized in the plant's body, and after that I discuss elaborately what is the function of the gibberellin. Okay, so here this is the biosynthesis, and you can see there is the stage 1, this is the stage 2, and this is the stage 3. Clear? Stage 1, the stage 2, and the stage 3. So the stage 1 that is seen in the plastid, the stage 1 that is seen in the plastid, the stage 2 that is seen in the endoplasmic reticulum. Okay, and after this happening, the actual biosynthesis is possible in the particular region. Okay? So what happened? You know all the what is IPP. IPP means isoprene unit. It is called the isoprene unit. Okay, so isoprene unit that is released from the pyruvate or the glyceraldehyde 3-phosphate. Okay, and then it converts to GGPP. What is GGPP? GGPP is the geranyl diphosphate. This is called the geranyl diphosphate. And this GGPP, due to the presence of CPS and the Kase enzyme. What is called the CPS? CPS enzyme is called, this is CPS. This is called the CPS. Yeah. The CPS is known as the end-copalyl diphosphate synthase. End-copalyl diphosphate synthase. This, in presence of this CPS and Kase, what is Kase? Kase is end-corin synthase. End-corin synthase. So, GGPP, in presence of these two enzymes, it converts into end-corin. Okay? End-corin, and this is the structure of the end-corin. And this situation or this total condition, you can say the end-corin's production is possible in the plastid. Okay, this is the plastid region. After the formation of the end-corin that enters into the endoplasmic reticulum, and before entering into the endoplasmic reticulum, here its conversion is possible. Okay, and it converts into Ga12ALD. Okay, Ga12ALD, and this is possible in presence of two enzymes. One enzyme is KAO, another enzyme is the KO. What is called the KO? KO is called the chorin oxidase, and KAO is the end-corinoid acid oxidase. Okay, chorin oxidase and KAO, that is the end-corinoid acid oxidase. In presence of two enzymes, it converts into Ga12ALD. Okay, and after that, Ga12, the formation of the Ga12ALD, it transfers to form the Ga12. Okay, Ga12. This after the formation of the Ga12, there are two pathways. Sorry for the disturbance. So here Ga12 is formed. Now in presence of Ga220 oxidase. Okay, Ga20 oxidase, Ga12 transformed into the Ga3. In presence of Ga3 oxidase is transferred or it converts into Ga4, and obviously in presence of Ga2 oxidase it converts into Ga34. Okay, so this pathway is called the non-13 hydroxylation pathway, and this stage 3 is occurring actually in the cytosol. So this what we learned here, the two pathways are formed. One pathway, that is the non-13 hydroxylation pathway, and another is the 13 hydroxylation pathway. Here from the Ga12, in presence of Ga13 oxidase, okay, Ga13 oxidase, it converts into Ga53. Okay, OX means the oxidase here. Oxidase. So in presence of Ga13 oxidase, the Ga12 converts into Ga53, and now this 13 hydroxylation pathway will start. What happened here? Ga53 that converts into the Ga20, and Ga20 that converts into the sorry disturbance. So here G53 that converts into G20, and G20 that converts into GA1, and GA1 that converts to form the GA8. This is the structure of the GA8. Okay, and here CH3 obviously. This is the CH3. Okay, CH3. So this is the structure of the Ga34, and this is the structure of Ga8. So what we learned here, the three stages. One is actually occurring in the plastid, another second stage that is occurring in the endoplasmic reticulum, and the third one, this part totally, that is occurring in the cytosol. And here the two pathways, after the formation of the Ga12 directly enter into the non-13 hydroxylation pathway, and in the next step Ga12 that converts into the Ga53 that enters into the 13 hydroxylation pathway, and the conversion is possible in presence of enzyme Ga13 oxidase. So here the hydroxylation process is occurring. Okay, from here Ga12, the transfer to or the conversion of the Ga12 to the Ga53 is possible due to the process of the hydroxylation. Okay, so this is the biosynthesis of the gibberellin. Okay. So I think you can understand it properly. Now we will learn about the functions of the gibberellin. Okay. Now we will learn that the functions of the gibberellin. Okay. So you can see here that the three functions I just elaborately describe you and write here. One is the stem elongation, and before going the topic, I obviously explain shortly that what is the stem elongation. Here I just elaborately describe it. What is the stem elongation? In case of plant, the application of the Ga to the dwarf mutant that restores a normal tall phenotype. Okay, if the plant is the dwarf, you can see that two types of plant. One is the tall, that the phenotype is TT and small tt. Okay, if this gene present, that means the plant is dwarf. Okay, and if this gene present, that means the plant is taller. So in case of dwarf plant, if we apply gibberellin from outside, then we will see that the dwarf plant that convert into the normal tall and phenotypic plant. Okay, so that means how this is possible? It is possible when we apply gibberellin that actually promotes the internodal elongation. Okay, internodal elongation by targeting the intercalary meristem. Okay, so I already described the two types of meristematic tissue. One is the apical, another is the intercalary system or intercalary part, or you can say the intercalary meristematic tissue. So what is the role of the gibberellin? When we apply the gibberellin, it actually the meristematic tissue that present in the intercalary or the internode. When the meristematic tissue in the internode, this is called the intercalary meristem. So what happened here? This gibberellin that promotes the internodal elongation by targeting the intercalary meristem and causes the increase in both cell elongation and the cell division. Okay, so it helps in the cell elongation and the cell division. Where which is the place? The place is the intercalary meristem. Sorry for the disturbance. Okay, sorry. So what happened? The stem elongation. Stem elongation actually the application of the Ga to the dwarf mutant that restores a normal tall phenotype. So what happened that when we apply the gibberellin on the mutant plant that has this type of genes, TT, small tt. If the capital TT present, that means the tall plant. Okay, and if the small tt present, that means the plant is the dwarf. So in case of dwarf plant, we generally apply the gibberellin. So what happened in that situation? Gibberellin that targeting the internodal elongation. And how the internodal elongation, you can obviously know what is the internodal part, the space between the two node portion in the stem body. So it just promotes the internodal elongation. How it is possible? That it just intercalary meristem. It target the intercalary meristem to divide the cells. Okay, and produce more cells. And in that way, it helps in the cell elongation. That means if the cell division is occurring in increased level in this part, intercalary meristem, then the elongation of the plant body is possible. Okay, and here the enzyme that is very important, that is the Ga3 oxidase. This enzyme is very important that catalyzes the conversion of the Ga20 to Ga1, to Ga1. And if the Ga3 oxidase that is absent in plant body, in that case the dwarfism is seen, because actually Ga20 is the precursor of the Ga1 and the conversion is possible if the Ga3 oxidase enzyme is present here. So if the Ga3 oxidase is absent here, that means the gibberellin cannot actually activate the cell division process in the intercavity meristem. Okay, so this is the stem elongation. Okay, and the next one is the seed germination. You always know that seed germinate in the favorable condition. Okay, that means the in presence of temperature, water, etc., and obviously the sunlight. These are the outer favorable condition or the climatic condition. And within the seed, you know that some reserve food is present. That is very important for the developing embryo. And what is the role of the gibberellin? You just you can imagine that gibberellin here play an important role. Okay, an important role. Well, how it is possible? There are one enzyme, some hydrolytic enzymes are present, and very important hydrolytic enzyme, that is the alpha amylase. Okay, this alpha amylase. This alpha amylase, actually, what is the function of this alpha amylase? This is synthesized and secreted from the scutellum. You always obviously know the one part in the seed, that is scutellum, and another part is the endosperm, and endosperm is the actually the part that is the reserve food content, or reserve food is present in there. In endosperm there are two layers. One is the starchy endosperm that contains the starchy grain, and another is containing the alluron layer that contains the protein bodies. So now what is the function of the alpha amylase, or the how it is related to the gibberellin? That alpha amylase is synthesized and secreted from the scutellum, and alluron layer and the hydrolysis starch chain Ga acts by inducing the expression of the gene for enzyme that is present there. What is the function of the gibberellin? That Ga, this acts by inducing the expression of the gene for enzymes. Okay, so if the Ga not activate the gene is not activated by the Ga, the enzyme that is called the alpha amylase is not synthesized. And if alpha amylase is not synthesized, that means the food that is present within the seed, that will be never soluble or absorbed by the embryo. Okay. So what is the function of the scutellum? Scutellum actually absorbs the solubilized food and that transport to the embryo. Okay. So gibberellin first that activate the gene, that is very important for the activation of the alpha amylase enzymes or other hydrolytic enzymes, and these enzymes actually help to solubilize the reserve food that present in the endosperm. Okay, and what is the function of the scutellum? The scutellum actually absorb this type of food and then transport to the embryo. Okay, so this food is very important for the embryo development. So in this way, seed germination, what is the role? The gibberellin helps in the development of the embryo. And the next one, in the next one is the flowering, or the sex determination of the flowering. Okay, in some plants, these are called the rosette plant. What are they called them? The rosette plants. Rosette plants are actually the plants. These are the examples: spinach and cabbage. You can say they haven't any flower. Okay, they haven't any flower. Why? Because there is a process occurring called the bolting. What is bolting? The before flowering, the elongation of the internode. Okay, elongation of the internode is occurring just before the flowering. Okay, and if the internode system elongated in the increasing level, you can see the rosette type of system of this type of plants. And this type of rosette formation is called the bolting. Okay, and this is possible due to the presence of the climatic condition. You can say the photo period, and I will describe about what is actually the photo period. So you just learn here the long day or short day type system. Okay, and the temperature. if cold or hot. Generally, you can see the spinach and the cabbage in the winter season, obviously. So, cold temperature is very important and long day is very important. So, without this, the bolting is not triggered. So, in absence of this, vegetable will not be grown. No, vegetable will grow by applying the gibberellin hormone from outside. And it actually promotes the bolting process to continue. Okay, and also it helps in the staminate flower in the cucumber. You can know what is the staminate flower. That means when the stamen that change to flower, this is called the staminate flower. So in case of cucumber, it helps to formation of the staminate flower, and obviously it suppresses the formation of this type of flower in the maize. Okay, so these are actually the functions of the gibberellin. I think you can properly understand about the biosynthesis and the functions of the gibberellin. Okay.